The cells of our body depend on signal transduction pathways to carry out different types of cell processes at the right moments in time to basically produce physiological responses to certain types of external stimuli. Now, even though these signal transduction pathways are very important to the functionality of our cells, these signal transduction pathways must be closely maintained and regulated by our cells. In fact, the inability of our cells to regulate and terminate these signal transduction pathways can actually lead to tumor growth and eventually cancer. And so in this lecture, what I'd like to focus on is discuss how different types of abnormalities in a signal transduction pathway can actually lead to cancer. Now, before we actually begin our discussion on the abnormality part, let's focus on how normal process takes place and how normally our cells terminate these signal transduction pathways. And to use an example, we're going to focus on the EGF signal transduction pathway, where EGF stands for epidermal growth factor. Remember, this is the pathway used by the cells that ultimately stimulates the growth and division of epithelial and epidermal cells. So let's begin by focusing on how this pathway actually takes place, beginning with the binding of the EGF molecules onto their domains. So we have two EGF molecules bind onto each one of these domains shown in purple, and once the binding takes place, these two monomers associate with one another to form a dimer, and that creates conformational changes in these two structures found in the cytoplasm. Now, these two structures, and by the way, this is the EGF receptor, these two structures actually contain tyrosine protein kinase domains. And once a conformational change takes place upon binding and the dimerization process, a cross-phosphorylation takes place. And the carboxyl terminal ends of this tail and this tail are phosphorylated by the active sites of these corresponding kinases. And once we form these phosphorylated residues, we have an important adaptive protein known as GRB2 that binds onto this section. And that calls upon another protein known as SOS. And what SOS does is it binds an inactive small g protein known as RAS. When RAS binds onto this structure, there's a conformational change that takes place in the RAS protein, and the GDP guanosine diphosphate is expelled, and the GTP guanosine triphosphate moves into that pocket. And once GTP binds, that activates that G protein we call RAS. And once RAS is activated, it moves on and activates a protein kinase we call RAF. And once this protein kinase is activated, it goes on to form these to activate other protein kinases we call MECs. The MECs, once activated, go on to activate other protein kinases we call ERKs. Now these ERKs can actually move into the nucleus of our cell. So we have this double phospholipid bilayer of the nucleus. These ERKs go into the cell nucleus and they activate transcription factors. These transcription factors then move on and express different types of genes that produce mRNA molecules, which then exit the cell, and they essentially are used by the ribosomes to produce proteins. The proteins are in turn used to basically build up the cytoplasm, build up the cytoskeleton, which basically increases the size of the cell, and that cell eventually is able to divide. And so in this process, the EGF signal transduction pathway stimulates cell differentiation, cell growth, and cell proliferation of two types of cells, epidermal cells and epithelial cells. Now, once this pathway actually carries out its specific purpose, how exactly does a normal cell terminate this process? Well, there are three major methods. Method number one is the fact that because we have a G protein involved, and G proteins have GTPase activity, what that means is they have a built-in clock that allows it to actually shut itself down following activation. So sometime after this has been activated into the GTP form, this green structure, the G protein, because it has GTPase activity, it is able to actually take a water molecule from the cytoplasm and hydrolyze the GTP back into GDP. And once it inactivates itself, this can no longer stimulate the rest of the process. And so this pathway essentially shuts down as a result. So cells can terminate the pathway by using GTPase activity of G proteins, and that is built into that molecule. Number two, these cells can also actually terminate the pathway by using a class of molecules we call phosphatases. So in fact, as soon as this pathway is activated, it also activates many different types of phosphatases. And what phosphatases do is they essentially remove phosphoryl groups that were attached by protein kinases. So for instance, we have the RAF, the MEX, the ERKs, and these two structures that act as protein kinases in this particular case. And what the phosphatases do is they essentially move onto the target proteins and they remove those phosphoryl groups that were placed by all these different protein kinases. For instance, these phosphatases can remove these phosphoryl groups here, and that essentially inactivates this part of the pathway, and so it cannot continue, and as a result, it is shut down. And so anywhere I have an asterisk, that basically means we're dealing with a protein kinase, and so these phosphatases can influence and shut down this protein, these proteins, and also these two structures here, which are actually part of that EGF receptor. And finally, we can terminate the pathway by inactivating that receptor of the pathway. And actually, we already spoke about one way by which we can inactivate it is by removing these phosphoryl groups. Another way is if these two ligands actually dissociate. When the two ligands dissociate, these two, the entire dimer basically breaks apart into monomers. And in that particular case, it is not as active as in this particular case. And so what that means is that will decrease the activity of this signal transduction pathway. So this is the normal way by which the pathway actually is terminated. But what happens in the abnormal case? How can an abnormality in each one of these cases, 1, 2, 3, actually lead to the production of tumors and eventually cancer? So let's focus on number 1. So we said our cells can terminate by using these GTPase activity G proteins. So this RAS protein has a certain gene in the DNA that expresses it. Now, let's suppose the gene is a normal gene, and that means this will be a normal protein. But what happens if that gene that encodes for this structure is actually mutated in some way? And let's suppose we mutate that gene in such a way so that this molecule loses its ability to basically hydrolyze the GTP back into GDP. And so if there's a mutation that takes place in the RAS gene, and the mutation basically destroys the GTPase activity of this G protein, then once the protein is activated, it will remain in the on position. It will remain active, and that means it will continually stimulate these processes, and the signal transduction pathway will continue the process of cell growth, cell division, and cell proliferation. So we see that one way by which a signal transduction pathway may malfunction is if a gene encoding for a protein that is part of that pathway is actually mutated. For instance, the RAS gene that codes for the RAS protein in the EGF pathway is the most commonly mutated gene that leads to tumor growth of epithelial and epidermal cells. So a mutation in the gene might produce a RAS protein that is incapable of ATPase activity, which basically means it cannot turn itself off after actually being activated. So this is essentially the off position, and this is the on position. And so if we have a mutation that destroys the ATPase activity of this structure, it will always remain on and always go on to activate RAF proteins, which will contain these processes that essentially lead to cell growth and division. Now, we see that these types of mutated genes that lead to the production of these cells that have cancer characteristics are known as onconogenes. So this is what an onconogene actually is. Now, let's move on to 2. So if there is an abnormality in this process, how can that lead to cancer? So we said that phosphatases are these proteins which are used to reverse the effects of protein kinases, and that essentially shuts down this process. Now, just like any other protein, in our chromosomes, in our DNA, we have genes that code for phosphatases. So for any given phosphatase, we have one gene that comes from the father and the other gene that comes from the mother. So let's suppose we have this homologous pair of chromosomes, and this is the gene that, let's say, comes from the mother, and this is the gene shown in blue that comes from the father. So we have the pair of alleles that code for that same type of, so this should be phosphatase, not phosphatase. Okay, so let's change that real quick. We have phosphatase. So we have these genes that code for phosphatases. Now, if these two genes are normal, then there really will be no problem. In fact, if one of them is abnormal, the other one is normal, usually the cell will be functional and will be able to actually shut down and terminate the process by using phosphatases. And by the way, because the phosphatases turn off the activity of these pathways, and that essentially decreases the likelihood that tumor growth will actually take place, these genes that code for phosphatases are also known as tumor suppressing genes, and the phosphatases themselves are known as tumor suppressing molecules because it's a result of these molecules that tumors do not actually develop. So phosphatases are called tumor suppressing molecules because they inactivate proteins and enzymes that drive that signal transduction pathway. And for this reason, we call the genes that code for phosphatases tumor suppressing genes. Now, what happens if both of these genes are for some reason inactivated, are for some reason mutated or blocked? So it can be some type of chromosomal abnormality, so a deletion, an insertion, a movement to some other chromosome, and so forth. So when both of these genes in the allele pair encoding for a specific type of phosphatase that inactivates some specific type of molecule in this process are actually knocked out or destroyed, that may lead to tumor formation because the cell will not be able to correctly terminate that process by using these phosphatases. So we know how abnormality can lead to cancer here as well as here. What about this final case? So we said that we can actually terminate the pathway by inactivating that receptor protein. In this case, it's the EGF receptor. Now, in some cases, we see that overexpression of these tyrosine kinase domains, so the tyrosine kinase receptors like the EGF receptor here, if the cell actually builds too many of these proteins and inserts too many of these proteins into the membrane of that cell, what that will do is increase the likelihood that the cell will activate the pathway at an inappropriate time. And so this excess number, the excess amount of these receptors, excuse me, the excess amount of these receptors in the cell membrane can basically lead to the production of these cancer cells. So overexpression of tyrosine kinase receptors can also lead to cancer of the epithelial cells. For instance, we have cancers like breast cancers, ovarian cancers, colorectal cancers, and so forth. So any place we have these epithelial cells, this can actually cause this type of cancer. So if a cell expresses too many receptors, the excess receptors may stimulate growth at inappropriate times. And actually, by studying this specific type of mechanism, we were able to create different types of drugs which act as anti-cancer, anti-tumor agents. And these drugs are essentially antibodies. So we were able to develop these antibodies in a laboratory that actually bind onto these EGF receptors. And once the antibody binds onto these pockets of the EGF receptor, that can basically inactivate these receptors. And by inactivating these receptors, that decreases the likelihood that this will lead to producing a cancer cell. And so one important drug that is basically used to treat individuals who have breast cancer is known as Herceptin. And Herceptin is basically used to bind onto a specific type of EGF-like receptor known as HER2. In fact, about 30% of the individuals who have breast cancer as a result of the excess quantity of this HER2 receptor in the membrane of their epithelial cells.